Hello everyone, I'm Xinzhang from the First Affiliated Hospital of Shantou University Medical College. Welcome to the online clinical histology courses. In this session, we will discuss blood and hematopoiesis. Blood is the only tissue that flows throughout our body. This red liquid carries oxygen and nutrients to all parts of the body and brings waste products back to our lungs, kidneys, and liver for disposal. It is also an essential part of our immune system, crucial to fluid and temperature balance, a hydraulic fluid for certain functions, and a special highway for hormonal messages. As such an important tissue, we call blood is a river of life. In this session, we will first go through the general characteristics of blood, include physical properties, components, and the functions of blood. Then, we will discuss the hematopoiesis and the hematopoietic system, and finally review two cases to understand the relationship between hematopoiesis defects and blood disorders. Blood is a specialized connective tissue and is the only liquid tissue in our body. Color range is between oxygen-rich scarlet red and oxygen-poor dark red. Total blood volume is equal to 7 to 8% of body weight, or around 4 to 6 liters. The pH value is between 7.35 and 7.45. The temperature is around 38 degrees Celsius, and other parameters are listed here in the slides. From our fundamental studies, we knew that human blood is composed of around 45% plasma and 45% cellular components. Plasma is straw-colored, sticky fluid. More than 90% is water, and solute include proteins, nutrients, hormones, electrolyte, and metabolite, and so on. The cellular component consists of three major cell types: red blood cell, white blood cell, and platelets. Among them, only white blood cells are complete cells. Red blood cells have no nuclei and organelles. Platelets are cell fragments. Most formed elements survive in the bloodstream for only a few days. Most blood cells are terminally mature cells, originate from bone marrow, and do not divide. Now we can give a description to blood according to its composition. Blood is a suspension of non-dividing end-stage cells of three types, namely red cells, white cells, and platelets. The suspending fluid is the plasma. Then, let's see the functions of blood. As we mentioned before, blood is so important because it has a number of functions that are central to survival. We classify it into three major functions. First, transportation. Blood carries oxygen and essential nutrients to cells and tissues, removes waste material such as carbon dioxide, urea, and lactic acid. Blood also transports hormones from one part of the body to another, transmitting messages and completing important processes. Another important function of the blood is its protective action against disease. As part of the immune system, white blood cells defend the body against infections, foreign materials, and abnormal cells. The platelets in blood enable clotting or coagulation of blood to minimize blood loss when the blood vessel is damaged. The third important function is regulation. Blood can regulate pH level, body temperature, and the electrolyte composition of the interstitial fluid to maintain homeostasis. Blood is one of the most regenerative and plastic tissues. Blood cells have a limited lifespan. Thus, these blood cells need to be renewed continuously. That is why hematopoiesis is so important. Hematopoiesis is the process of formation of mature blood cellular components from immature precursor cells, which occurs during embryonic development and throughout adulthood to produce and replenish the blood system. Human hematopoiesis is a very complex process. Blood cells are one of the first differentiated cell lineages generated in the embryo. It initiated in the yolk sac during the 3rd week of development, which is very beginning of embryogenesis, because the embryo requires red cell for the transport of maternal oxygen to support its growth and development, which means the hematopoiesis develops together with embryo, span before and after birth stage, and functions throughout the whole life. Thus, it undergoes sequential, spatial, and temporal changes during development. To discuss the ontogeny of hematopoiesis, we can divide the process into 2 parts, prenatal and postnatal stages, which means before and after birth. Hematopoietic regulation in the human fetus differs greatly from that in an adult. In an adult, hemostatic maintenance is the prime function of hematopoietic regulation, whereas in the embryo, the fetus' constant changes characterize all phases of hematopoiesis. Knowledge of developmental hematopoietic regulation helps clinicians to interpret postnatal hematopoietic data. This picture shows us the overview of hematopoietic sites transition during development. We can see it changes from yolk sac to placenta, which are the extra embryonic sites, then transit to intra-embryonic sites AGM, fetal liver, and finally to the bone marrow. These slides show us the hematopoietic sites change and contributions to hematopoiesis during development, before and after birth. Here, we listed the hematopoietic sites and time during the whole development process. Then, next, let's look at the development of hematopoietic cells. Hematopoietic stem cells are the seeds for all lineages of blood cells, originate from yolk sac and finally reside in the red bone marrow, constitutes about 0.5% of total bone marrow cells. Similar to small LC in shape, they have 2 properties: multipotency and self-renewal, means they can generate all lineages of blood cells and can copy themselves to maintain the pool of HSCs. This picture shows us the whole process of hematopoietic stem cell to generate all types of mature blood cells, from long-term HSCs to mobilize to short-term HSCs, then differentiate to common lymphoid progenitors and common myeloid progenitors, then to erythrocytic progenitor, megakaryocytic progenitor, granulocytic progenitor, and lymphocytic progenitor, and finally all types of mature blood cells. Other than hematopoietic stem cells, the hematopoietic microenvironment, termed stem cell niche, ensures hematopoietic homeostasis by controlling the proliferation, self-renew, differentiation, and migration of HSCs and progenitor cells at a steady state in response to emergencies and injuries. We will take bone marrow, the major hematopoietic site of adult, as an example to discuss the organization of hematopoietic microenvironment. The bone marrow is organized as hematopoietic islets, composed of hematopoietic cells, stromal cells, and extracellular matrix. Stromal cells include reticular cell, fibroblast, macrophages, endothelial cells, and adipocytes. They support hematopoietic stem cell to control the hematopoiesis. Stromal cells express and secrete extracellular matrix glycoproteins and adhesion molecules to allow the fixation of hematopoietic cells within the bone marrow microenvironment, control migration of hematopoietic cells within and out of bone marrow. They also produce hematopoietic growth factors and cytokines, which are glycoprotein hormones that regulate the proliferation and differentiation of hematopoietic progenitor cells and the function of mature blood cells. Here in this slide, we listed all the hematopoietic growth factors and cytokines required in the hematopoiesis. Now we have gone through the process of hematopoiesis. Many molecules and cells are involved in this complicated process. Any defects or abnormalities may cause quality and quantity change of blood cells and lead to disorders. And so far, the etiology of most of these disorders are still not known. Now, we review two cases to understand the relationship between hematopoiesis and blood disorders. Case number 1. A 37-year-old male, a primary school teacher, came to visit the office and complained of weakness and fatigue. He spilled and gave a history of petechic and hematuria. CBC tests showed all types of blood cells are lower than normal, but blood smear showed normocytic red blood cells. Then reticulocyte count showed less than 1%, and bone marrow biopsy showed more than 70% are yellow marrow. And the diagnosis is aplastic anemia. The second case is a 39-year-old female who has the history of fibrocystic breast disease, seen for routine workup prior to breast biopsy. Physical examination found moderate splenomegaly. CBC test showed a high number of white blood cells. Bone marrow biopsy showed markedly hypercellular with increased megakaryocytes, and cytogenetics test found translocation between number 9 and the number 22 chromosomes, which is known as Philadelphia chromosome, and 90% of chronic myeloid leukemic patients carry this mutation. This mutation will cause uncontrolled myeloid cell proliferation, and patients was diagnosed with CML. Now let's summarize this session. We discussed the general characteristics of blood, include blood components and functions. Then we discussed hematopoiesis during development, which occurs in spatial and temporal distinct sites. We also discussed interaction of hematopoietic stem cells with microenvironment and generate all lineages of mature blood cell. Finally, we review two court cases to understand the relationship between hematopoiesis and the blood disorders. That's all for this session. See you next time.